And while I'm talking, we're sort of explaining how this class is going to work. If you haven't done it yet, log into Blackboard and download the Lecture 2 in-class notebook. And if you need help, this is what these sticky notes are for. The red sticky note, you can attach it to your laptop and it says you need help, and that should get our attention. Come around. This has worked well at a bioinformatics boot camp that was run earlier this year, I believe. And if you're doing okay during the activities that I'm going to explain, put the green sticky note up there. And so before we get started, we have several tutors and TAs who will help you out, who will monitor the sticky notes. And so I want to introduce them here for you so you know who to look for. I wanted to actually stand up and tell us who you are.
Hey, guys, I'm Brett. I'm a sixth year student now in the Rockford's lab in the computational systems project.
Tell me about it. I'm the computer systems student in the Rocks.
I'm Steven. I'm a second year HSG student in Leading's lab.
I'm Jeanette. I'm a third year student in molecular genetics and genomics in Jeff Williams' lab.
I'm Drew. I'm a genetics student in Joe Corpo's lab.
All right, so during the activities, if you have put up a red sticky note, if you need to get somebody's attention, wave your hands. They're going to come around and help you out. All right, welcome to the first computational lecture of this course. So as you've figured out by now, surely, we're going to alternate statistics lectures with computational bioinformatics lectures. And the purpose of this is to help you learn how to do the things you learn about in the statistics portion. Because a lot of your calculations, you don't want to necessarily do them by hand the way you had to do them for the first homework, because in some cases you're going to have a lot more data to work with. So in this class we're going to learn how to program so that we can implement these statistical methods. That's the main goal of the program. There are a lot of other areas of bioinformatics we could talk about, but in this class we're going to stay focused on the statistical aspects. And we're going to use, as you've seen by now, we're going to use the programming language called Python, and we're going to use a particular way to use Python called IPython Notebook. We chose Python because really that's I would say that's the scientific computing language of the future right now. That's where everybody's moving. You may yourselves have worked with or know other people who work with other languages in science. There's Perl, which is what I started out with. A lot of people use R for data analysis. Some people use MATLAB for modeling, engineers in particular. Python has the advantages has the capabilities of all of these languages, but it's also easy to learn, easy to use, and it's quite powerful. So that's why we chose Python. All right, let's see. Finally, let me explain a little how this class is going to work. I'm going to talk for just a little bit, not too much. And then, since you learn best by doing, we're going to break out into in-class activities. So maybe I'll talk for 5-10 minutes, and then you'll do an activity for a little while. We'll reconvene, we'll talk about the activity, I'll introduce the next activity, and then you'll attempt to do it, hopefully complete it, and so on. So today, we're going to have three breakout activities. During the activities, you can work in small groups, work with your neighbor, or by yourself. Get help from wherever you need it. And I don't want to bore you with a PowerPoint lecture, but since we're going to be working on the computers, I will be demonstrating things up here. I have a few slides just to sort of illustrate some of the things that I'm going to talk about. So I guess the first point I want you to take away from this first computational class is that programming is going to be essential for your career in biology. Starting your graduate training in the year 2015 going forward, programming is going to really be critical in the future. That wasn't always true in biology, but it certainly is now. So why is it true? If you're not going to be a computational biologist, well, even if you're not a computational biologist, you're likely to work with data that requires some computation. This may be hard to read, and you're not supposed to really read it. here's the raw output of a next generation sequencing run. This is the kind of file that comes there. So a lot of data is based on sequencing, working with files like this. Here's another sort of, and again, you can't read it, but it's just sort of raw data output from mass spectrometry. Some of you in biochemistry may end up doing mass spec experiments. If you want to be able to work with your data, this is what it's going to look like. And even if you don't do sequencing or don't do mass spectrometry, but you do microscopy, this is some data from one of my friends and colleagues, Arjun Raj at UPenn. People these days are quantifying their microscopy images. So here's an image looking at RNA transcripts, and then it's quantified over here, and that all involves programming. So even if you are likely to require a little bit of programming in the work that you do, but even if you don't, you might want to use somebody else's data that requires it. So basically, in the future in biology, data is increasingly quantitative, and it's increasingly large scale, and it increasingly involves sequencing information. So to work with all of this data, you need to know how to program. All right. Second, here's a tip as you learn how to program. And as you learn to explore, as you learn about genomic data, you should really explore. Okay, that's the best way to learn how to do it. You're not going to learn how to program simply by reading a book or sitting through a few lectures. To really gain these skills and to become familiar with bioinformatics, genomics, you have to explore. And there are all sorts of great free, open, online resources. This is just an example of one sort of package that we'll talk a little bit about later that comes with Python called NumPy. It contains all sorts of great tools for doing statistical and mathematical calculations. They have some links. They have this Getting Started page. So just navigate to the page. I'll give you the link later. Click on a link and start reading. So you really should explore. The other places to explore are genome databases. This is the NCBI webpage is good for more than just PubMed. There's a ton of data up there. And you just sort of search around their site, and there are all sorts of places you can start. Right at the top left here, it says using genome, using their genome database. Help, that's the first link. So I encourage you to explore. Click the links. You won't break anything by clicking links and exploring the data. Okay. So those are sort of, so that's the reason for why you're learning programming, and a tip for how you can learn to program and work with genomic data you should explore. Let's see, we'll get there in just a moment. I'm going to, so what we're going to do in today's class is we're going to learn how to do something that sounds simple in principle, but then when you think about how to do it, it might be tough. So what if somebody asked you, or what if you decided you wanted to know for some project, what is the average length of all 3' UTRs in the human genome, of all genes in the human genome? It seems like a simple question. Maybe you can Google it, maybe you can't find it. How would you calculate it by yourself? Well, that's what we're going to do today. If you want to know the average length of a 3' untranslated region of a transcript of a gene, we're going to show you how to do it. So what we're going to do today is step 1, we're going to download some genomic data, the data we need for it. Step 2, we're going to bring that data into the IPython notebook where we can analyze it. And then step 3, we're going to calculate the lengths of 3' UTRs, and then we're going to calculate the mean and the variance and some of the summary statistics that Zach talked about last time. So basically these 3 steps sound simple, but there's a lot packed in there, so hopefully it'll be interesting. All right. Okay, so this first part now, we're going to go get genomic data. And the first thing you should know about genomic data, or any sort of high throughput quantitative data, is really, it is stored in what's called a flat file, or a plain text file. So this is key idea number 1 that I want you to take away. All that fancy high throughput data, it's stored in files that you can open up in any text editor. It's not like Microsoft Word where it's a proprietary format. There's no bold, no italics, it's just a plain text file, which is critical because that means it makes it easy to share and easy to load into IPython and work with, or whatever programming language you want to use, as you'll see. So here's an example. I already showed you an example of what comes out of those Illumina sequencers. The output that you get when you send your samples in for sequencing is basically these raw reads. So one read here is delineated by a plus that separates your record. There's some information about the read in the top, and then the actual sequence of the read is the 3rd line at the bottom. Notice it's all plain text, no formatting there. All this fancy data you see in genomic databases, I'm just trying to see how this looks here. You can see this is a shot from the UCSC Genome Browser. It shows you a portion of a human chromosome. It shows you areas that indicate these are genes, coding genes, all sorts of genomic data, there's conservation and things like that. It looks all fancy. Underlying all of that, plain text files. That's all it is. This is ChIP-seq data from here, and this basically gives you the chromosome, the coordinates of the chromosome, and the number of reads under the peak, and a little bit of other information. So that's the key idea. All this fancy genomic data comes in plain text files. And you can get it in lots of places. You can get it at your favorite model organism database. So all the standard model organisms have a database. Here's the yeast genome database. They have a download page, and you can go download the plain text files of the data. You may be familiar with these databases. You can go look up your favorite gene, and you can use their nice user interface to gain information about that. But if you want to work with genomic data, you go to the download page and download some files. Flybase has it here. Wormbase has got it. And they even give you information about what is in their latest release of data. If you're interested in mouse or human or clinical data, here's an example of the ClinVar database, a database of clinically relevant human genetic variants. It's filled, they have an FTP site and other things. Plain text files. And so if you want to work with this data, it's easy. You don't need fancy software. All right. So that's it for the PowerPoint lecture for this first part. Now we're going to do activity 1. So in activity 1, we're going to go to one of these genome databases, the UCSC Genome Browser, and we're going to download a table of all human genes and their coordinates. So remember, we want to calculate the lengths of 3' UTRs. So we're going to calculate download a table of gene information. And the way this first activity is going to work is I'm going to bring up my browser on the board here, and we're just going to follow along, because that's the best way to do this. We'll just do it live. If you run into trouble, put up a sticky note, wave your hand if something's not working. All right. Any questions so far? We all set? All right. All right, so pull up your browsers here. Pull up a web browser. Hopefully you can see. Might be a little hard to see what I'm doing, but you can navigate to genome.ucsc.edu. All right, we pull up the genome browser here. This is the UCSC Genome Browser. This is, I think, if you work in human or mouse genomics, this is one of the most valuable databases of genomic data. Let's just take a look here. So you can see it on your screens. This may be a little small to see up here, but we're just going to look at some of the features here. Look, they have genome for the Neanderthal genome data. Click on that and see they give you links to all sorts of things there. They have ENCODE data, if you're familiar with the ENCODE project, if you want to get some of that experimental data. A lot of people are excited about that data and want to somehow correlate their experimental results with the ENCODE data. You can find that here at the UCSC Genome Browser. There's all sorts of information. We're going to use a useful tool that's up on the left here called the Table Browser. The Table Browser is a great tool because it pulls data from different sources and it creates a plain text file for you that you can then download and analyze. All right, so is everybody in the Table Browser here? We all good? All right, so as you can see, there's a help link at the top using the Table Browser. There's a user's guide and all that. So if you want to explore, there are links here to explore. What you see here at the top is our query form. This is where we tell it what kind of data we want. And so some of this is self-explanatory. If you scroll down under using the Table Browser, it explains all of those inputs there. It can seem a little overwhelming, and it is at first, but most of this stuff we're not going to worry about too much. Actually, I'm going to reset this to the default settings so that you're seeing probably where you're at. Okay, so let's start at the top. Let's choose what we want. So we want to get a gene table from the human genome, right? So you can choose a clade. We're going to choose mammals. This is not really phylogenetically accurate, obviously, right? Mammals are vertebrates and all that, but this is just sort of a useful grouping of data. You pick your genome, right? There are a lot of genomes to choose from here. Now here is the assembly. This is actually critical. The human genome sequence gets updated periodically as data improves, as they revise things. So the data you are interested in sometimes may refer to a specific, may not refer to the latest assembly. So when you're going to analyze data and correlate perhaps your data with data from another experiment, you need to make sure you're choosing the correct assembly. And more important, when you write a paper that uses this data, you need to specify which assembly you used. All right? We're going to choose the data we want is from the February 2009 assembly. So go ahead and change that field. All right. Now we're going to look at this next line here. This is sort of, now we're narrowing down. We've chosen our genome, we've chosen our assembly. Now we're going to choose the kind of data we want. And they've grouped data into useful categories. You can see there's mapping and sequencing. That's if you're interested in sort of the sequencing of the human genome, or perhaps, the quality of the sequence or whatever other genome you're interested in. There's regulation, gene expression, variation. We're going to focus. We want a gene table, so we're going to do genes and gene predictions. Now, now we've chosen roughly what kind of data we want. Now we're going to choose specifically where that data comes from. So there are many options for gene predictions here. We are going to choose, look, there's a lot here. This can be overwhelming, and it is. Somebody who comes here isn't necessarily expected to know everything on this list. You have to go out in the literature, you have to be familiar with the data, you have to know a little bit what you're looking for. We're going to choose the GenCode genes, version 19. This is from a consortium called GenCode, which has a set of gene annotations and gene predictions in the human genome. All right? So we'll skip these over here. Now we're going to choose exactly the table we want from the GenCode gene data set. You can see if you click on this, they give you several options. There's the basic, there's the comprehensive. You can pick pseudogenes and other things. We're going to choose the basic today. How do you know what you chose? All right. How do you know what's actually in this basic table? All right. Here's something important. This button's going to be important for your homework here. So if you want to know what's in the table, you click this button, describe table schema there. It's kind of an awkward name for it, but this is actually filled with incredibly useful information. Here's the data you're going to see here. So let's look at the top. So it tells you schema for GenCode genes version 19. It tells you what it is, and it gives you a little information about what's in there. So it gives you the name of the table, a description, a gene prediction with some additional information. It's not very specific, last updated. Now look here at the top, the row count tells you how much data is in there. So we're going to download a table with 99,900 items in it. Now this tells you, this table here tells you actually what's in the table. So the field name are basically your column headers in the table, and it gives you a description. So bin, this is just something useful for the genome browser to use. It gives you the official name of your gene, at least in the GenCode data set. You can see we got chromosome, strand. Actually, let's work across here. Let's, for example, look at the chromosome field. As you go across, it gives you an example of what you're going to see in your table. It gives you an identifier of what type of data it is. And this is happens to be for SQL. It tells you whether it's a number, a character, whatever, something like that. If you want to know exactly what the whole range of values is in there, you can click on this link and it'll tell you, and then here's your description. So there's chromosome. You can see we have, going down the table now, transcription start, transcription end. Okay, and now here's an example of the table you're going to download. If you look down there. All right. Everybody with me so far? It's pretty straightforward, right? All right, we're going to hit the back button now. We're done looking at what's in our table. Now we want to finish downloading. All right. So we're going to skip a few of these things here. They're really not critical. Right now we want to actually let's look at region briefly. We're going to do a download for the whole genome. There's actually a really useful tool if you slide over to define regions. You could actually create a file of just the regions of the genome that you're interested in and extract the data from that. So you can paste the name of the chromosome and the coordinates, or you can upload a file, and instead of downloading data for the entire genome, you just get the parts you're interested in. Extremely useful. We're not going to use it today, though. Some of these other things get a little confusing. We're not going to worry about them too much right now. Let's go down to output format. All right. We're going to choose what we get from this table. That table had a lot of fields. We're not really interested in all of them. So we're going to select, let's open this up, we're going to select some of the fields. Some of the other options give you other possible outputs. It's these top couple here that are going to give you the plain text file that of the type we're interested in. So let's choose selected fields from primary, and notice now it says and related tables. So now this gives us the option to draw in data not just from the table we looked at, but from elsewhere. All right, let's go see where else we can get our data from. So click on Get Output. I find this button a little confusing. When you click it, you kind of expect to get your output right away, but it actually brings you to, in this case, another form. There. Here's our table, the header names of our table. Remember, now we're going to choose a few. So follow along with me carefully here. This is what we're going to download to use in the rest of our activities. We're going to skip the bin. We're going to choose the name field, chromosome field. We want to enter the strand, start it, transcription start site, transcription end, coding sequence start, coding sequence end. Let's do the exon count. I don't even know exactly what SPORE means. It's something related to gene prediction and, you know, the more familiar name of the gene. All right, we got that. Everybody selected those items. Now look, we asked to draw from additional tables that could be linked with this table, so it's giving us one choice. There's one table we can choose from. So we're going to click this table and we're going to see what's in that table. And this is a little non-intuitive, a little weird here, but because this, on the top you're seeing headers from one table. On the bottom you're checking for an entire table. So it's not the best user interface, but it works. So let's click Allow Selection from Check Tables. Now it's showing you the fields of this table. And if you wanted to go back to the main page and look at the table schema, you could look at all that and know what this is. But we're only interested in one thing here, the biotype of the gene. It's going to tell us what kind of a transcript is it. Is it a coding sequence? Is it a link RNA or miRNA or something like that? Okay? All right, we got that. We've checked the items we want in our table. So here we go. Now we're really going to get our output. So click Check Output or Get Output. There we go. And we have our table here. There's your plain text file. Does everybody see it? Everybody got one? Any problems? Nope? All right, great. This is what we're going to work with. So now save it. It should work in your browser if you just hit Save, Command S, or whatever browser you're using. If you need help, raise your hand and save it. And let's, so you should have created a class folder during the primer. If not, create one now. Wherever you, just so you know where it is, wherever you're going to load it up. And call it, let's just give it a name. Let's call it Gene Table. I don't know, I annoyingly put in 2 capital letters in there, and that's kind of in the text, but name it whatever you want. Anybody need help? If you need help, now's the time to get it. Yeah, I'm gonna.
Does it matter the format we save it in?
It should be a text, a plain text file. What formats is
Is
it?
that page source or web archive?
Web archive. So you may need, in your case, are you going to Chrome or
There's actually
oh, weird. It's not. When you save the web page. Try this. How about either download it or you can hit select all. Yeah, this is yeah, and then just save that. Yeah, good. Yeah, it depends on your browser and your setting. In my case, I can just save it. Another option is to hit Command A to highlight everything. Oops. Sorry, this is running slow.
If you go to the table browser, there's like a little dialog box.
Is it giving you an output? Oh, did you find a better way to? Great. Okay.
So if you go here, you can just type in the file name.
Oh, sweet. Excellent. I've never even used it that way, so good. Yeah. All right. Here, let's show everybody. Let's see. You should be able to type in your file name, I guess, here. Let's try this, right? .txt. Now that we've entered all of this, we have a plain text file selected. Get output. Let's see what it does. In this case, it takes me here. Is it? Oh yeah, and then it downloads. Okay. Well, I should have had to do that step before. All right. Is everybody good now? You learn something every day. All right. That's the end of activity 1 here. We have our file. Everybody good? All right. You should be able to see it in your class folder here. I'll just sort of illustrate one last thing. You can open it up in a plain text file. There it is. You got your column headers. So I'll show you one thing here. You are perfectly free to edit things in this text file. For example, you could. These are long column headers. Don't edit the column headers all yet. We're going to do something in Python, but I'm just going to give you an example here. Right? We could just change that to name. It's a lot more intelligible. Save your file. Boom. That's the beauty of a plain text file here. All right. We have our data. We have our genomic data, our table of almost 100,000 rows. Great. Any questions so far? There's obviously a lot there. I encourage you to go and explore the genome browser. All right, that was activity 1. Now we're going to get ready to go into activity 2. So I'll briefly, and then after that we'll take a break. So now we're going to use Python. We're going to use Python in a particular way. So Python is interesting. There are 2 ways you can use Python. You can use it to write actual programs, and those are written, again, as plain text files. And you can save those files and then just run them again and again from your command line. This is particularly useful if you want to write a program that will process something you want to do over and over again. That will process your next generation sequencing data. Every time you get sequencing information back and you have those plain text files, you can write a script and that'll process it. And the other way is interactive. And that's pretty much the way we've been using it right now in IPython Notebook. Although IPython Notebook can also run scripts. So here's an example. You're not expected to read all the details of script mode. This is coming out of our lab here. This is basically a program for doing some modeling on gene expression data. It's just written as a plain text file. The colors actually come from the fancy text editor that can tell what parts of Python are commands and this and that. But really, it's just a plain text file. That's a script or a program. But we're going to use it in interactive mode, which is the way you've already used it. And that's great for exploratory data analysis. Kind of like a calculator or whatever. You can do something and immediately see the result and look at your data and figure out what you've got. All right, so that's key idea number 2. The first one was plain text files. All right, now let's go to key idea number 3. So in the homework, you wrote functions for calculating means and variances, all that. Which is great. You should know how to use the basic Python functions. But one nice thing about Python is that lots of other people with, in some cases, with substantial expertise in statistics or engineering or math have written useful, free Python tools. And this is a critical part of how you use Python for data analysis. You're not going to program everything yourself. You're going to use some of the great tools that are out there. Let's see here. And so I'm just going to, you can think of these packages of extra tools basically as toolboxes of power tools. And there are a few main toolboxes that we use over and over. They're called, NumPy, SciPy, Matplotlib. We'll see what some of these are. IPython is what you're using with the notebook. So these are extensions to Python. And these were, most of these at least were downloaded with your Anaconda installation. You don't even need to go find them. So for example, Matplotlib, you can make fancy graphs. It would really be a pain to learn how to program, or even not learn, but just to program one of these nice looking graphs by yourself. And there's a tool to do it. I'm listing really the main critical Python packages here. NumPy contains lots of mathematical routines, really important for statistical analysis. SciPy is kind of an extra supplement to NumPy, calculating integrals and other sorts of things like that. Finally, here's the toolbox we're going to use today. It's called Pandas. And one way you can think of this is it gives Python kind of like spreadsheet capability. This will be like Excel on steroids here. This is really a great tool for data analysis for working with our plain text data table. Let's see. And so the way you use a toolbox is basically two steps, or what use a tool. You first have to open the toolbox, okay? So you have to, once we're in IPython notebook, you open the toolbox using this command called import. You give the name of the toolbox as, and then you can give it a nickname, which will save you some typing. So for example, we're going to type import pandas as, and there's its nickname, PD. And then you use the tool, and there's this dot notation. You've already used a little of it in your homework with the append function. Basically, you use the nickname of the toolbox, or in other cases, it'll be your data your variable here. You name the tool after the dot, and then you give the dot certain arguments, a file name, numerical values, a variable, whatever it is. All right. So that's what we're going to do. We're going to basically use this toolbox to import our data into IPython notebook. So you should all have downloaded this sort of lecture 2 IPython notebook from the Blackboard page. Everybody got it? Any issues with it? All right. Now, if you want, you can close this or open a new one. Now, so go ahead and launch IPython. You can use your Anaconda launcher. You can go to the terminal, type IPython notebook. Go ahead and launch it. If the computer's old like mine, it might take a while. That was fast, okay? Come up on your dashboard. Now remember, this is all basic stuff. You know how to do this. Navigate to your course folder. There are faster ways to do this. I'm just doing this the slow way. I have too many folders. Class folder, there we go. All right, and there's lecture 2, IPython notebook. Click on it, and you're up and running.
Mike,
Yeah.
I might remind everyone to double check that next to lecture 2 on that little browser, they can see the text file that they downloaded.
Yes.
If you can't see the text file on that page, you should raise your hand now.
Yeah, you'll run into trouble there, right? So there's genetable.txt. You should see the one you just downloaded right in there. Everybody see it? Hey, okay, great. Because we're going to need that. Yeah, there we go.
And take note of what its name is.
Yeah, see its name there. You can always go back to the dashboard if you need to remember. All right, so here we are. So now I'm going to turn on the lights, break into small groups, do it by yourself, ask for help. This should be kind of self-explanatory, although there will be some challenging parts here. So this is the activity. Go ahead and read and work your way through it, and ask for help as you need it.